Hi, everybody. Welcome to our first lab of Module 3. Today, we're going to be working with DNA extraction. We're going to do a virtual PCR, which is a polymerase chain reaction. And then we'll be doing electrophoresis virtually on a gel chamber. Now, the first part of this lab, we're going to be extracting the DNA from a couple of strawberries. So I wanted just to first go over the introduction to this whole lab. So the purpose of this lab is to demonstrate to you that the simplicity of biotechnology. I mean, we're essentially doing biotechnology in our kitchen with just a couple of household products able to extract DNA. Now, most of the techniques that are employed today in a laboratory where they're doing, crazy gene editing and stuff like that, most of those techniques were developed from normal like laboratory utensils and stuff. There wasn't too many innovations in terms of like machinery that the novelty of machinery that aided in biotechnology. Now, we have PCR machines today, and we'll talk about that when we get to PCR, that do the three-step process of changing temperature for us. But in the past, we just have 3 different water baths at different temperatures and just move our sample over, for 30 rotations. So anyways, some things that we can do with biotechnology, which is kind of cool, is we can take segments of DNA from one organism and we can cut and splice them into the DNA of another organism. And you probably saw that in our first lecture where I showed you the pig that had glow-in-the-dark feet and a nose, and then we also had like a glow-in-the-dark tobacco plant. So that's called a GFP protein, and those come from glow-in-the-dark jellyfish, and they're able to extract that gene and put it into the jellyfish. And so it's pretty cool what we can do with, we can knock out genes, we can silence genes, we can turn off genes, we can move them around. The thing about genetics, the thing just about playing with genes in general that makes it kind of tricky and somewhat dangerous is that not all genes have one product. Some genes have an entire pathway of products that they actually switch on or switch off. And so when you start taking genes in and out, oftentimes you'll have meaning more than one, polygenetic effects when you just manipulate one gene. And so that's when it becomes really tricky, because it's like, oh, this one gene causes some disease, and so you're like, okay, let's get it out. And you take that gene out, and then it's like, oh wait, it caused this other disease to take it out, and then you're kind of like, well, now what do we do? So there's a lot of nuance involved when we're doing genetic studies, and so yeah. So I already gave you the objectives. We're going to isolate DNA from strawberries. We're going to show you how to make lots of copies of DNA, specifically with certain regions of DNA using PCR. And then we're going to show you how we separate those fragments in a process called agarose gel electrophoresis. And of course, that's virtual as well. You're going to need your Adobe Flash Player. Okay, well, let's jump into DNA extraction. I'm not going to read you this paragraph. Hopefully you've read it. Hopefully you've read it before this, or you can pause right now and read it. But part 1, DNA extraction. So we're going to basically isolate some DNA, and I'm going to skip all that stuff. So I want you guys just to read that. I could read it to you, but that'd be kind of boring. So let's do our real DNA extraction. Let's just talk about what kind of stuff we'll need and why. Okay. So what we need is our coffee filter. What I've done is I've taken 2 small mason jars. These are the 2 what are these? The 2 empty glasses or plastic cups. These are the 2 empty glasses. One of these, you're going to mix your lysis solution in. So that'll have like your salt and your soap and your water in it. And then the other one is going to be where you capture the filtrate. So after you've mashed up the strawberries to release the DNA inside of them, you will then filter it through this. And so I'm just showing you, I have this small, it's probably like an 8-ounce mason jar. And I was able to take the coffee filter that came in our kit, and I just kind of put it around the inside there. And then I used the rubber band that was holding our transfer pipettes together. I just use that rubber band around the edges, and that'll make it easier when I go to dump the strawberry mush inside of it. Okay, so we've got those 2 jars. That's what I'm using. I've got 2 plastic bags. One of these we're gonna mash stuff up in. The other one, I forget what I do with that one. I don't know. We'll see when we get there. I've got a tablespoon and I've got a teaspoon. I've got a transfer pipette. Now, I don't have any wood stirrers or coffee stirrers, but I do have a transfer pipette from the kit, and that will work just fine for picking up the DNA. I've got half a cup of water already measured out. I've got a couple of strawberries. I had to borrow some dish soap from my neighbor because I'm running low. And then I've got some salt here. And that is about all the supplies you're going to need. Okay? All right, so let's get started. So we've collected all the supplies we need. Oh yeah, I almost forgot. The isopropyl alcohol. The reason I don't have it out in front of me is because it's actually in my fridge. If you're just now starting this and you're like, oh crap, it's not cold, just throw it in the freezer. It's not gonna freeze, but it'll get cold. And the colder, the better for the isopropyl. All right, so once you're ready to start, go ahead and grab your strawberries. And it says you can use 2 medium or 1 large, so I'm just gonna grab 2 medium strawberries. Now we don't want any of the green stuff on them. And so what we're gonna do is just take off the green and kind of pick the leaves off. And then that little stem, I kind of picked that out. And so what we're left with is just strawberry. Now you can chop it up, but dude, like if your strawberries are ripe, they're gonna smash up pretty easy. So I'm gonna open up the baggie. I'm gonna put that strawberry in there. Let's pick off the green from the other strawberry. Kind of dig out its stem, and then put that in there. Yeah. All right. We put them in the bag, and so what we're gonna do is we're gonna mash these up for about 2 minutes. And like I said, strawberries are pretty mushy. You can chop them up if you're feeling like you don't got the grip strength for this kind of stuff, but it ain't that bad. One thing I do recommend is getting the air out of the bag. That's gonna make it easier to smash them up. Okay, so we got our strawberries. We're just mashing now. And as we're mashing, we can read ahead and see what's next. So we want to completely crush these. And then we got to ask the question, so why are we crushing these up? Why don't we just stick the strawberries in the solution and try to extract their DNA? What we're trying to do is we're trying to maximize the surface area that our lysis solution will come in contact with. So consider this, right? Like if I just stuck 2 strawberries in a salt and soap solution, the only DNA that would come into contact with that would be on the cells that are on the surface of the strawberry. But when we mash it up like this and get it really mushy, I've released all of those cells, not all of them, but I've released a lot of them into solution. And so now we have a lot of individual cells, a lot more cells that will come into contact with that solution, thus helping us with our DNA yield. All right. Smash, smash, smash. All right. And once they're smashed up, it doesn't have to be perfect, but you know, the mushier, the better. So mine are pretty dang mushy. I'm just gonna set that off to the side. Now what we need to do is we need to make our DNA lysis, our extraction liquid. So we're gonna need 2 teaspoons of soap. So I got my teaspoon. I'm gonna do 2 teaspoons of dish soap. 1 and 2. Doesn't have to be perfect. You know, most of these are just kind of like general guidelines. So 2 teaspoons of that. 1 teaspoon of salt. I should have done the salt first, but it's all good. I'll just pour it in the spoon. Hopefully I don't make a mess. There we go. 1 teaspoon of salt. Set that off to the side. And half a cup of water. Okay. Now you're going to want to mix that up. So I'm going to use the teaspoon that I just used to measure the salt and the soap. I'm going to mix that up. And now it says take 2 tablespoons from this glass that we just made. And we're going to put them in the bag with the strawberry. So let's think about what we're doing here. Right, why would we do that? So if you remember from the introduction, in order to get to the DNA itself, we have to get through let's see, the outer membrane and then the nuclear membrane. And then there's a membrane on the inside. So the nucleus is a double membrane. So there's actually 3 membranes we got to get through. And what the soap does is, because soap is kind of a, what do they call it? I forget the name of it, but it breaks down lipids, and so it'll break down our lipid membranes. So I'm going to take 2 tablespoons of that. We're going to seal them up in the plastic bag again. Again, the more air you get out, the easier it is to kind of mush it. Otherwise, you might accidentally pop the bag open and you have to start over and it'll be a big old mess. Okay, so we're going to smash, however you want to call it, for another minute. And we're not going to try to make too many soap bubbles. We just want to get the soap in contact with the membranes. All right, and so if you haven't done it yet, the next step is placing the coffee filter over the glass. So you're going to stretch the coffee filter over the glass. I tied it with a rubber band. That's really helpful. If you don't have a rubber band, you can always use like string. All right, so I'm gonna smash this for a little bit longer. All right. And we're just smushing. All right. So we had the salts in there, and we had the water, and we had the soap. Okay, so we're ready to do the next step. All right, so we placed the coffee filter. We're gonna take this strawberry stuff and we're gonna squeeze it into the clean cup. So this is the part that takes a little bit of patience. So I'm just gonna drop it in the coffee filter. And what this is gonna do, it's gonna help us get out all the residual big chunks, all of the cell walls and things like that. I'm just gonna throw that bag away. So it's basically gonna help us get all the big chunks out. Now, we can sit here for a few minutes and just kind of let it drip. You can also kind of stir it with your finger if you want. It's kind of gross, but it feels gross. But ideally what we want to do is we want to squeeze out and just end up with the liquid in the bottom of the jar completely. So I'm going to pause the video. You guys go ahead and do that. Just remember what we've been doing. So we have the salts, we have the soap, we have the water, and then we have the alcohol. And the soap is breaking down the membranes. The salts are essentially distracting the water from the DNA. And then we're using the ethanol to get rid of the salts and just separate them from the DNA itself. And so that's the whole purpose here. Like I said, I'm going to pause this until mine's done draining. You can do the same. And then we'll get our ethanol out of the fridge or out of the freezer. Okay, I got my ethanol from the fridge. We're just going to pour it down the side of the glass. We're just going to pour it down the side of the glass and let it sit. We're not going to stir it in. And it should sit right on top of your liquid. And then it said within a little bit of time, we should start to see white, wispy stuff forming on the top of our extraction. And if you can guess what that is, that's your DNA getting pulled into the ethanol. So remember that DNA is, has that phosphate backbone, which is electronegative. And so it's going to dissolve in a polar substance, and so that's what the ethanol is. The ethanol is drawing, it's attracting the DNA out. So yeah, we should start to see it forming. And then once your DNA is on the surface, feel free to take your micropipette and you can start to pick it up. It's going to take a few minutes, so I would let it sit for, I'll tell you in a second. I'll start a timer, and what I'll do is when mine looks good, I'm going to pause the video, start a timer, and then when mine looks good, I'll start the video and I'll tell you how long to set your timer for. All right, I lied. First, let's cover the questions that are, now that you're an expert in DNA extraction, answer the following questions. So what I do is I put my jar in the fridge, because the colder the ethanol, the better. And as it sits at room temperature, it's going to warm up and be less effective. So I put mine in the fridge. All right, at a minimum, what materials do we need to isolate our DNA? Well, first we're going to need DNA. So the source DNA is a good one. We're going to need to get some sort of lysis buffer. So a lysis buffer would be something that has something that dissolves liquid or lipids, something that dissolves lipids, and also has salts in it. And so we're going to need salts, soap, water, source DNA, and then finally ethanol. And so the ethanol is going to help to get the salts out and separate the salts from the DNA. And so those are the minimum that we would need. Are strawberries eukaryotic organisms? Well, I can tell you this. Anything that you can see with your eyes is going to be eukaryotic. Think about that. Anything that you can see with your eyes, visibly, to the naked eye, is eukaryotic. How do you know that? Well, bacteria is prokaryotic. It doesn't have a nucleus, and it's not multicellular. So there are some single-celled eukaryotes that you cannot see with your naked eye, but every single multicellular organism, which is ones that you can see with your naked eye, are going to be eukaryotic. And so if strawberries are visible, they have to be eukaryotic. And so that means that they have membrane-bound organelles like a nucleus. And so what are the structural challenges that we might encounter when extracting DNA from the cell? Well, let's think about it. A structural thing we might encounter is a cell membrane. Another thing would be the nuclear envelope and then the nuclear membrane. And so you've got 3 phospholipid bilayers that you have to get through in order to get the DNA. And so that would be structural challenges. Number 3, why did we crush up the strawberries in step 2? I mentioned this earlier, and then what do we accomplish? Well, we're increasing the surface area that the lysis buffer will come into contact with. If we just dunk the strawberries in the lysis buffer, they wouldn't. They would only extract DNA from the very surface of the cells, and you wouldn't even. It would only extract DNA from the cells on the surface of the strawberry, and it might not even be enough DNA to see with the naked eye once you do that. So what do we accomplish? That's what we accomplish. Oops. I spilled salt. Better throw it over my shoulder. Sorry, someone was calling me trying to refi my house. Okay. We crushed them, we're done. Okay, so number 4, why do we add salt to the DNA in step 3? Well, the salt is going to preoccupy the water molecules, so the water molecules are not going to dissolve things like big structures like cell membranes and proteins and things like that. And so the salt distracts the water molecules so that the bigger stuff that we don't want sinks to the bottom. That's all it does. So the salt is sort of like keeping the water molecules in handcuffs so that the only thing staying in the solution is going to be water, the DNA, and the salt. And then that's why we add the ethanol, is the ethanol takes the salt out, and then, well, the water in the ethanol draws the salts out, and then the ethanol itself pulls the DNA into the ethanol, and then that way in that ethanol layer, that's where we get our DNA. Okay, and so that answers the questions to that. I'm going to check my thing in the fridge. Oh, we got more questions. So my DNA has been in the fridge for about 5 minutes, and I still want to leave it a little longer. So what happens when we add the isopropyl alcohol to our DNA solution? Why were you not allowed to stir or mix the solution? Well, the isopropyl alcohol is going to sit on top of the lysis solution, and it's going to draw or attract the DNA into the alcohol. I think I might have called it ethanol on accident. We use ethanol in the lab at school, but they gave us isopropyl. It's the same thing, small alcohol molecule. Why were you not allowed to stir or mix the solution? Well, because it would dissolve into the lysis one, and then there would be no separation. And so we don't want to stir it. If we stirred it up, it would homogenize the solution, and you wouldn't be able to. Yeah, the ethanol or the isopropyl alcohol would dissolve the DNA, sure, and but it would also dissolve it inside of the lysis solution, and so you wouldn't see it. You need it to sit on top so it attracts just the DNA to the surface. If you stirred it in, it would mix and homogenize with the lysis solution, and then we wouldn't be able to see the DNA. Is DNA water soluble? That's question 7. DNA, let's see. Let's think. So DNA has a negative charge. It's kind of like an anion in that sense. So it's got a negative charge because of the phosphate groups. We know that other ions like salts dissolve readily in water. So I would say DNA is water soluble based on that. So yes, and it has a negative charge. All right, the next thing that you guys will do after this is you'll do your polymerase chain reaction. Definitely read that story about the surfer. This like really hits home for me because it's like this reminds me of grad school. As I'd be trying to solve a problem and it would be on my mind all the time, and I would just be thinking about it. And so this guy's driving his car when he finally like got this idea for how to amplify certain regions of DNA. So definitely read that. And then what you're gonna do is you're gonna go to you're gonna click the links on your computer, and you will go to watch a video on how PCR works. The video is pretty helpful and it's pretty short. And then go to the Rutgers Virtual PCR Lab, and you'll have to click that link as well. And so while you're running through your PCR reaction and doing the lab, go ahead and answer those questions, like what you put into the PCR tube, what is 95 degrees Celsius in Fahrenheit, how many copies are created in the thermocycler. You can answer all those questions on your own pretty easily just from running through the virtual PCR program. And then same thing for the gel electrophoresis. It's the same idea. Like you should be able to answer all those questions really easily. And then finally, the last thing you would have to do is maybe leave your glass in a fridge, leave the glass in the fridge until you go through the virtual labs and answer those questions, and then go get your glass out, take your transfer pipette and pull out your DNA and take a picture, upload the whole thing, and you're done. All right, guys, I'm gonna pause it. All righty. And mine didn't make a whole lot of DNA. I'm gonna try to hold that up and not drip it on my keyboard. Mine did not make a lot of DNA. It didn't extract a lot, but what's hanging from my pipette right there, that is definitely DNA. So maybe you guys will get more. I'm excited to see your pictures and see how much you got. All right, we'll see you for the next lab. I almost forgot something on the virtual labs. Using their pipettes, sometimes you have to hold the click down on the mouse. Like you have to click the mouse and then hold it down, like hold the button down on the mouse while you pick the pipette up out of the solution, and then you let the click off the mouse when you're trying to squirt it to wherever it's going. It took me a little bit to figure that out. I just thought I'd share that. All right, we'll see you guys for the next lab.